All right, so this section is going to be on non-Mendelian genetics. We've been going over Mendelian genetics in the past section. This is specifically going to be looking at non-Mendelian genetics. These play by some of the rules of Mendel's P plans, but not all of the rules. So these are a little bit more complex and have like exceptions, we like to call them, to what Mendel found in his P plans. So without further ado, let's get into non-Mendelian genetics. The first here is incomplete dominance, which is also known as intermediate dominance. And this is when the heterozygote or the heterozygous genotype will have a different phenotype than the parents. If you see here, we have these flowers that are called snapdragons. They're red, white, and then when you mix them, they appear pink. Now, this looks like the blending hypothesis because we have the white and the red mixing to make pink, but it's not. The reason why this is not the blending hypothesis is when we cross two pink flowers, I'm going to show you this next, we don't just get more pink flowers. You'll see we get some red and some white. If the blending hypothesis was true, you just keep on getting more pink flowers, and that's not what happens here. But in incomplete dominance, you don't have either allele being dominant or recessive. They're equally dominant, or I shouldn't say they're equally dominant, but there's just no dominant or recessive. So in class, we do this Punnett square, and you can see here when we cross a white and a red, we get all these pink flowers because when we have two R alleles and two W alleles, that makes a white and a red. But when we have one of each, that makes a pink. Now let's say we cross two of these pink flowers. You will get some more pink flowers, yes, but you will also get red and white flowers, showing that this is not the blending hypothesis. This is just a different type of genetics or just different type of inheritance. These are chickens called Andalusian fowls. They also express through incomplete dominance, where you can see we have the black chickens, we have the white chickens, and in the middle we have these chickens called the Andalusians. They're the blues. They're not really blue. They're like I said, bluish, grayish color, but they're in between the black and the white chickens here. All right, next is codominance. Codominance is where both alleles are going to be equally dominant to each other. These are a type of cow called roan cows, and you can see they're expressed by the capital letter. So big R is red, big W is white. You can see here we have this roan cow, which has both R and W. Now, you might ask, Mr. Fradov, is WR, does that create a roan cow? Yep. As long as it has one of each or the heterozygous, it's going to be that roan cow. And you can see how in the roan cow, this is a little bit different. It shows both colors. So it's both red and white. It's not a mixture. It shows both red and white. Let's look at the Punnett square that makes a roan cow. So we usually do these in class, but you can see a red cow and a white cow. The R stands for red, W stands for white, produce these roan cows, which are both red and white. A type of codominance, or an example of codominance, is human blood typing. I always like going through human blood typing because I think it's pretty interesting, and students can really relate with this because they have their own blood types. But a person's blood type is represented by something called multiple alleles, which we have multiple alleles here. So you have multiple, not just one or two. And it's also codominant. So you can see we have three different alleles, A, B, and O. Now, usually I write this as capital IA, capital IB, and I for the three alleles. But in this case, we in like biology, we just do the ABO blood system. It's a little bit easier for us to recognize. In humans, blood types A and B are equally dominant to each other. That means if you get it, you're going to have it no matter what. Both types of blood, though, are dominant to O blood. So you can see here we have both types being dominant to O. That gives us four different phenotypes. So we have A, we have B, we have AB, and we have O. And you can see here the different genotypes. To give you an example, I usually have my AP students understand this concept of the IAI or IAIA. I just have my biology students focus on the ABO. So A, if you are blood type A, you can have two different genotypes. It can be AA or it can be AO. B can be BB or BO. AB, there's only one genotype. It's AB. O, there's only one genotype. It's OO. Notice how we have the same alleles here, AA. This is the homozygous form of type A blood. AO is the heterozygous because they're different. Remember that the hetero means different. That's the heterozygous form of A blood. Same with B. We have BB, that's the homozygous. We have BO, that's the heterozygous. So again, our genotypes, just like I mentioned, AA for A blood, AO for A blood, BB and BO for B blood. AB is the only type of genotype that produces AB blood, and OO produces O blood. I'm not going to go through the examples, but what I want to talk about is blood typing and what you can receive from like somebody with AB or BO, whatever blood. If you have A blood, you have these little flags, I call them. They're antigens, but they're little flags on the blood cells that say, "Hey, I'm A blood." If you have B blood, your your antigens are B antigens. They're a little bit different. If you're AB blood, you have both A and B antigens. If you have O blood, you don't have any antigens. Now, back to the A blood. So again, you have these A antigens. These are little flags that say, "Hey, I'm A blood," and your body recognizes them that way. But you also produce this antibody called B antibody. This antibody attacks B blood. It's not going to attack A blood. It's going to attack B blood. If you have B blood, you have, again, these B flags on your red blood cells. You have A antibodies. These attack A blood. If you have AB blood and you have both these flags on your red blood cells, you don't have any antibodies. And if you have O blood, you have both A and B antibodies. These antibodies protect us from like getting somebody's wrong blood in our system, but if you have AB blood, you don't have any of those antibodies. If you have O blood, you have both of those antibodies. This can create some different reactions when people are giving blood, so we have to be careful what type of blood we give to who. Type O blood is a universal donor. So since I'm going to go back here, since they don't have any antigens on their red blood cells, they can be given to anybody. Because if you see this O blood, it can be given to A blood or A blood over here because they don't have any antigens. They're fine. So we know O blood as the universal donor. You can see this chart. You can give it O blood to everybody. AB blood is the universal receiver. It's because they can receive from anybody because they don't have any antibodies to react with the blood. So O blood, universal donor, AB blood, universal receiver. And you can see there are different phenotypes of A, O, AB, and A, B, AB, and O blood. There's different amounts. We usually see O blood being the highest amount and AB blood being the lowest amount. All right, so lastly here for blood typing, this is the Rh factor. There are more antigens. All you need to know is the Rh factor. If you are positive, you have the antigens. If you are negative, you do not have the antigens. If you look here, we have the Rh positive blood, Rh negative blood. So positive people can be positive for their genotype or positive negative. They can be homozygous for positive or heterozygous for positive. The negative people have to be negative. So in this case, positive blood is dominant over recessive blood. Again, more example types. And I like to show this image at the end here. You can see like difference between A, B, and O. If you have positive blood, you have the Rh factor. If you have A blood, you have the A antigens. If you have B blood, you have the B antigens. And if you have AB blood, you have both of the A and B antigens. All right, the next type of inheritance is going to be called sex-linked genetics. And these sex-linked genes are going to be found on your sex chromosomes. So remember, males have an X and a Y chromosome, females have an X and an X chromosome. So males only get one X, females have two X's. And we see more sex-linked genetics being found on the X chromosome just because it's larger and it has more genes. You can see here the X chromosome has about 1,500 genes. The Y chromosome only has about 80 genes. So again, any sex-linked gene is just any gene located on the sex chromosomes. Let me go back. In humans, most sex-linked genes are going to be found on that X chromosome, just like I was saying. There's just more genes there. Now, when we're talking about these sex-linked disorders, males typically get these sex-linked disorders more often than females because if we inherit one of those X-linked genes that maybe has a disorder on it, we automatically have it. Females need two of them usually to get that disease or disorder, so it's harder for them to acquire two compared with us where we just need one. I would say one and done, but males have it, females, you need two. Carrier. This is an individual who has one copy but doesn't have the disorder. Females in this type are going to be the only types of carriers we have because, again, males, if you get one, you already have it. Females, if you get one, you cannot have it because you're going to be a carrier. So you can see here the difference between affected females and the unaffected males. You can see unaffected female, X big H, X big H. Carrier female, X big H, X little h. You can see how there's that one little h there. She won't have the disease, but she will carry the disease. And you can see X little h, she's an affected female. She has to get two of them. You can see it's a lot harder to get these, both of these little alleles. Compared with males, you either don't have it or you have it. So in this case, X big HY is an unaffected male, X little HY is an affected male. Color blindness is a great example of sex-linked genetics, where men get this much more often than females. It's because we only get one of those. If we get one of those chromosomes that has color blindness attached to it, we automatically get it. Females need both. Females can be colorblind, but it's a lot more rare because they have to inherit both of those genes. This is a colorblind test. I always remind students, don't freak out if you can't really see this number. This number is 12 here. The colors on these with these images on my computer aren't the greatest, but this is a colorblind test to help you determine, or help a scientist or a doctor determine if you are colorblind. I always like showing this as hey, this is what the colorblindness looks like if you're not colorblind, and what the person then sees. All right, so I go through some examples here, and yep, we're going to stop for there. All right, another type of non-Mendelian genetic is polygenic inheritance. And like the name implies, poly means many. And this is where several alleles are going to contribute to a certain phenotype. So it's not there are only one allele. So like in Mendelian genetics, we saw one allele or one genotype producing the color of the flowers or how tall the pea plant grows. This is where several alleles are going to contribute to the phenotype. And there's a large amount of phenotypes that you can see here. Human height is an example of polygenic inheritance. This is where several factors or several genes are going to contribute to the height of a person. And there's a large distribution. So we have people that are really, really tall, really, really short, and right in the middle. What we call these curves they're called bell curves. And we can just see that the men here on average are a little bit taller than the females, but there still is a large, just large distribution of human height. I would like to show this as a person, on this graph here. This is, I think, the 1996, Connecticut State University. You can see this distribution, the bell curve in the human height. Skin color is also determined by polygenic inheritance of a certain protein. We're going to look at that more in class. I'll try to attach it to the Google Classroom, but you're going to see here that this has evolved in humans over thousands and thousands and thousands of years due to the location where certain civilizations really evolved. And we're going to talk about this more in class, but this is also a polygenic inheritance. Oh, this is where non-Mendelian genetics stops. I will make a short podcast on pedigrees next.